Good afternoon, fellow researchers. Thank you for coming to my presentation. Let me introduce myself. I'm Elvan Wiyarta from Universitas Indonesia. In this opportunity, I would like to share and present my research about the potential of Valeria macrocarpa leaves ethanol extract to inhibit the expression of Ki67 in mouse distal colon after dextran sodium sulfate induction. In this presentation, I would like to share my research starting from introduction until conclusion. First, I would like to explain the background of this research in just one simple word: inflammation, which is the basic process of various deadly disease, including ulcerative colitis or UC. UC has gained importance over the past few decades because it is a part of incurable chronic inflammatory disease. This could happen because of some various factors, ranging from genetics to abnormal tissue structure that ultimately cause prolonged inflammation. The incidence and the prevalence of UC in the world are also very high, as we can see here, especially Western Europe and Southern Asia. This chronic disease, aside from affecting patients' quality of life, also requires high medical costs. If not treated, patients with UC can increase the risk of colorectal cancers up to 20 times. Over the past few decades, a lot of research has been done to find effective treatments for UC. Until now, the recent therapy goal is to treat inflammation as soon as possible, which include aspirin and corticosteroids as the drug of choice. However, these drugs have several side effects, ranging from bone depletion to male infertility. Other therapies that are currently being developed are herbal therapies derived from plants that is natural and inexpensive. Not many people know that here, in Indonesia, we have native plants called Mahkota Dewa or Valeria macrocarpa. Its anti-inflammatory properties is thought as an alternative therapy for UC. Until now, research on the development of PM as anti-inflammatory therapy is still very minimal. Therefore, we conduct a research on PM using Valeria macrocarpa leaves ethanol extract or PMLEE. PMLEE is administered into mice colon tissue that has been modeled UC using dextran sodium sulfate. DSS is a pro-inflammatory agent that's widely known to cause prolonged inflammation. Furthermore, to assess PMLEE relationship with UC, the Ki67 protein was chosen as a marker. Ki67 is the tissue biomarker that has been commonly used to assess the inflammatory process. This research is expected to be able to provide insight into the anti-inflammatory mechanism of PMLEE, as well as the basis for the development of PM as a potential alternative therapy for UC. Next, I want to explain about material and methods that we use to conduct this research. First, we prepare the PMLE by homogenizing Valeria macrocarpa leaves into fine powder. Then, the powder was soaked in ethanol, filtered through, and thickened, then ready to be administered as PMLEE. In this research, we used BALBC mice as our experimental animals, and divided into several groups, ranging from normal to negative control. All groups received 2% DSS administration except normal group. Then, each group received different kind of treatments based on their group. We have 6 groups which consist of 5 mice each. In total, we have 30 mice. These mice are then grouped using simple random sampling, 5 mice per group. The experiments will occur for 3 weeks, which in the first week they will receive 2% DSS, except normal group. Then in the following weeks, they will receive treatment based on their group. At the end of the 3rd week, all mice are terminated and their colon were collected. The colon then undergo tissue handling, which consists of fixation, paraffin embedding, and sectioning, which produce histology slide as the results. The slides then undergo immunohistochemistry process, starting from deparaffination, rehydration, antigen retrieval, blocking, primary antibody using Ki67, secondary antibody, enzyme labeling, chroma again using DAB which will produce brown color, counter stain, dehydration, mounting, and finally visualize under microscope and take the picture of each slides. These taken images then used for the Ki67 quantification process. For the quantification, expression of Ki67 shown by the brown color density. To quantify the brown color density, we use new technology called Automated IC Profiler. The software will process the image based on the pixel value threshold and generate optical density score as the result. If any of you want to know more about this technology, I would be very happy to explain it in the question sections, or you can just mail me. In order to be analyzed, ODS need to be converted into H-score based on this formula. Finally, the H-scores are then analyzed using ANOVA and SPSS. That's our method. Moving on to the results of this research. Qualitatively, the expression of Ki67 can be seen by comparing the brown color density in each field of view. As we can see here, normal and positive control clearly show low brown color density. Negative control and PMLE 100 somewhat show high to medium brown color density. And PMLE 200 and 300 somewhat show medium to low brown color density. But these are all too subjective, because different person may have different perception on how light or dark a brown color is. For more objective results, we're moving on to the quantitative results using H-score. This is the summary of all H-score in each group, and this is the mean difference between group based on Temhain test. These results are what I am going to explain in the next section, which is the discussion. First, let's take a look on the ulcerative colitis model. As we can see here in this graph, there is a significant difference between Ki67 expression on the negative and positive control. Why could this happen? Ki67 expressions in the negative control could be very high because of DSS administration. As I already mentioned before, DSS is a pro-inflammatory agent which will increase Ki67. Furthermore, several factors also modulate the effect of DSS, such as dose, in which in these results we use 2%, duration, in which in this research we use 7 days, and mice strain, in which in this research we use BALBC mice. On the other hand, the Ki67 expressions on positive control could be very low because besides given DSS, positive control also given aspirin, which has been known as an anti-inflammatory agent based on this mechanism. Also, in this research, we find some interesting findings. Surprisingly, the normal group did not show significant difference with either positive or negative control. This could happen for several reasons, including high variability, human errors, and system errors. As we can see here in this diagram, there are several ways to prevent these errors to occur. In order to avoid human errors in data collection process, pathologists need to ensure each scan photo meets quality standard. In fact, according to Afner et al., crucial role of pathologists lead to the collection of meaningful and reproducible data before being processed by IMERI. We should also do system check to prevent system error, and we check data for some inhomogeneous results. Then, finally, let's take a look on PMLE effect in this research. As we can see here, all PMLE groups except 100 milligrams show significant difference with negative control. There is also significant difference between PMLE 300 and 100. This implies dose-dependent manner because different dose yields different Ki67 expressions. Both of these effects could happen because of several compounds in this amazing plant. Let me provide you with some studies. Some studies, like Mariani et al., believe that hydroxyl benzophenone glucosides that contain in Valeria microcarpa has slight anti-inflammatory effect. But latest study from Diantini et al. Shows that benzophenone glucoside was not able to significantly reduce the proliferation of human cervical cancer cells, which indicate that inhibition of Ki67 expression by PMLE might not be caused by the substance. Interestingly, the latest study from Seich et al. Shows that flavonoids, especially epigalactocatechin galat, is known to inhibit the Ki67 via PRBE2FDP pathway. This study emphasized the role of flavonoids to inhibit Ki67. Well, guess what? Surprisingly, Valeria microcarpa does contain flavonoid. In this study, 0.3%. Furthermore, according to Kusmardi et al., the type of flavonoids contained in PM, camphorol, was thought to be one of the factors that influenced the decline of Ki67 expressions, based on Kin et al. Camphorol is also known to inhibit Ki67 via NF-kappa and JAK-STAT pathway. Overall, these studies strengthen the PMLE findings in this research. As a conclusion, PM is a herbal plant that has been widely studied as having anti-inflammatory effects. This effect was also related to the expression of Ki67 in cells. as in the study it was inversely proportional. The relationship between PM and Ki67 still need to be investigated. In addition, the dose of PM is also a determining factor for Ki67 expressions and need further research. Research related to the use of other inflammatory markers on UC besides Ki67 need to be known and need to be done in order to understand anti-inflammatory properties of PM. I think that's all from me. Thank you for your attention.